We're good to go. All right, so like Alex said, if you guys can navigate to the link that I put in the Slack, this will help you change profiles to the R Studio. So once everyone's here, let me know. There's like general thumbs up. All right, so let's press start my server. And now you should have an R server or like R Studio window. Does everyone have this? Yeah. Okay. Who here has used R before? Okay, so a decent number of you. But I'm still gonna go over like R basics just for those of you who maybe are not super familiar with it. So this is R Studio, which is an interface to R that you can use. It makes coding in R, I think, a little bit more accessible to people who are not computer scientists, which I think is really nice. So you have this console over here, which is where you would run your commands. So like if I want to see what 2 + 2 is, it'll tell me. Over here you have your global environment. Yeah.
Can you zoom in a little?
Oh, yes, yes. Is that better? I can't hear. Oh, okay, cool. All right. Yeah, so we have the console over here. And then over here is our global environment. So any values that you might like have here, your environment should be empty. Mine just has this leftover variable from something else, but they'll be stored here. You can see like what data type they are. If it's like a list, you can kind of like access it and see the dimensions and that sort of thing. If you want to clear your environment, there's this broom here, which can be helpful when like your RAM gets full. And then down here, you have this window with has all your files. And then if you have any plots, they're going to show up here. These are the packages that are available to you. You can install them or like load them by just hitting like selecting them. But there's also a way to do it in the code. And if you need any help in R, there's this really great help resource. And so you can look up like any topic. Another way to look up help for a function, say I don't know how to make like a data frame. We'll figure out. You can do question mark and then the function that you want to look up. And it will pull the help file for that function. And this also, I think, works like offline if you're like, I don't know, for some reason off the grid in coding, but it's just really nice to like always have this at your disposal if you're confused about anything. A lot of times there'll be like examples at the bottom. But yeah, so that's like just basic introduction to R Studio. And then we're gonna navigate over to files. Then into the OceanHackWeek tutorial folder. US Thursday, and this SDM's in R folder. And I'm gonna pull up this R markdown file. So if y'all could open that, if you want to follow along. All right, so this is called an R markdown file. It is a way that you can code in R that's somewhat similar to a Python notebook. And this is really great because you can integrate both text and code simultaneously. And you have a lot of options about like how it will render. And so this particular document's rendering as an HTML. And so if you all like after this week want to go back and look at a fully like just run version of this with like the text and stuff, you can go to this HTML and it should like render in a internet browser for you. There's some stuff at the top, like you can specify a title, just all sorts of stuff, like you can get more advanced with that. There's this first chunk here, which is this knit R thing. This is like always in an R markdown. This basically is like calling the function, like the package that will like knit the file for you, which means just like put it to like the output HTML. Yeah, I'm gonna start like clicking through just a brief intro to R. Just sort of like how things work before I get into more of like an R application, like a real world example of how you would use this. So first we're gonna start loading our libraries. These are similar to like packages in R or packages in Python. So if you wanted to install a library and you didn't have it, You're not gonna be able to do that here, but the way you would do that is with this install that packages function, and then you put like whatever package you want. So like if I wanted to install this like dplyr package, you just put it in a string, and then this will install it. This up here will load those libraries. So we're loading this tidyverse, which is a data science library that has a lot of functions for sort of like neatly processing data. We're also going to load this R-OBIS package, which will apply to the species distribution modeling portion of this tutorial. Biomod 2, which again applies to the species distribution modeling portion, and this Terra and raster package, which handle more like gridded data types. So before we get into the species distribution modeling, I just have like a few R basics in this first chunk here. When you are working in R Markdown, you can run like a whole chunk by itself. So like here I have, like if I wanted to like load all my libraries at once, for example, I'll just hit this play button and it should have like run all of this. So you don't have to like click through line by line, which is kind of nice. Yeah, so going down here, this is just like how you assign a variable in R. This is the preferred way, this like backwards arrow. But you can also do this equals. Also, this is like too basic for you guys, like let me know and I'll move on. But just some like basic math operators, pretty similar to like what you have in Python. This just kind of shows how you would do like an equivalence with the equals. And now I realize that I'm not seeing what I can. I'm just gonna switch my so if you want to like switch how your RStudio window looks, you can go to this global options. Then I'm gonna go to like pane layout, and I just want my console over here so it's easier to see side by side. Yeah, so now like we can see what code is running. Yeah, so we have like x times 2 equals 4. This is like just showing that both of these mean like the like to square x, but also just shows like the equals sign, which is the same as Python. There's like just inbuilt, you know, numbers and like pi and that sort of thing. And then just like a super useful tip that has like really helped me get better at R programming is using something called a pipe. And so if we have something like I'm going to create this vector here using this C, which basically is just going to like take all these numbers and put them together. So if we look at X, it's just like a sequence of those numbers. This does the same thing, but this here will compute the mean of X, and this here will also compute the mean of X. Does anyone know like what's happening there?
I have another question. How are you running the lines?
I'm so sorry. I totally was like gonna tell you guys. So what you do, if you're on Windows, you're gonna do Control Enter. If you're on Mac, you're gonna do Command Enter. Alternatively, if I wanted to run this line, I could just go up here, hit run. Selected lines. So sorry. But yeah, does anyone know like what this little like line here is doing? Yeah, so basically there's this like pipe operator in R, which allows you to tack a bunch of like functions onto each other. So this will take whatever is before the pipe operator as the argument to the function after the pipe operator. And so what this is doing is taking X as the argument that's gonna then go into this mean function. And so this might not seem like super useful in this particular example, but you can imagine if you're doing a lot of like data frame manipulations and you want to like add a column or like rename something, you can like pipe a lot of these together and then like do a lot of like operations simultaneously instead of having to go back and do them one by one. So it makes you like a little bit more efficient. Does that concept like make sense to everybody? That's like a little bit of a tough one for people sometimes. Okay, cool.
I have a question.
Yeah.
Would you just like comma after mean and then like add your other?
Yeah, that's a great question. I do have more examples of this later, but what you would do is you would just add another pipe. So if I wanted to do mean and then I don't even know, median, which makes no sense in this case.
I think you're asking if you had multiple arguments to a function.
Oh, multiple arguments to a function.
No, but this also is.
Yeah, so this is how you would kind of pipe multiple things together, but if you have multiple arguments, like for example, in mean, you can like specify to remove the NAs from the calculation. You would just put in those arguments as if that first argument does not exist. So basically, R can see that X is going to be that first argument, and then whatever is given after that, it's going to know is after X, if that makes sense. So if we do this, yeah, this is going to run exactly the same as we were going to do this. I know, again, this is like not a very helpful example, but helpful ones coming up. So next I'm gonna like go into data frames a little bit. I made this like really fake zooplankton data set. I don't really know what I was thinking here. But you can make a data frame using this like data.frame. And this is similar to like the pandas data frame structure. And they can do this in multiple ways, but what I've done here is basically you specify the column name as a string and then like whatever values it's gonna have. So in this case, I've like, actually just gonna show you what exactly this is. So if we view this, it's made this table of like 3 rows with all these columns and then these values. And then there are like multiple ways to access columns in data frames in R. This is one way of doing it. If I wanted to pull out like just the genus column, for example, I would do that. There's this really nice feature where if you like type your data frame object and then this $sign, all the column names will come up. And so like if you start typing like A, because I want abundance, and then hit tab, they'll just fill in. And so that can be really nice when you like don't totally know maybe like what the names are, you have like a ton of columns or something. And then if you wanted to access multiple columns, this is one way that you could do it by you're like accessing columns in the data frame a little bit differently. So if I wanted to get abundance using this alternate method, I would have to do this. I like never used this. I don't know if that's actually good. Yeah, so this does the same thing as like this up here. It's gonna give us these values that are like printed down here. It just looks a little different. Anyway, this is how you would access multiple columns, for example, using base R. Then if you wanted to add a column, you could just like type a new name after the $sign and just put whatever data you wanted there. Like if you wanted to modify a column, this kind of just goes to show that you could do like some data frame operations here with like subtraction and stuff. And then there are, like I said, there's this tidyverse, which is like this data science universe with like all these really great packages that make data manipulation a lot more intuitive, in my opinion. And so I'm just gonna like go through similar stuff with like what we just went through, but with dplyr. And so this is how you would add a column in dplyr. So not using that $sign notation like we did right here. Instead, if I wanted to add like, what is the sampling method, for example, I could use one of those pipes. So I'm taking that original data frame and piping it to this dplyr mutate. This dot 2 colons just indicates that mutate is coming from the dplyr library. And then I would just type in the name of my new column. It doesn't have to be in quotes, and then the values. So if we do this, we see that that has been added. And then you can also like filter the data frames if I only wanted to select like the Calanus species. Yeah, there's only one of them because these are terrible data. And then this is an example of like piping multiple functions together. So this is going to take our zooplankton data. It's going to add that sampling method like we did previously. It's going to filter for the genus Calanus, and then it's going to select only the genus, abundance, and sampling method columns. So now we have data that looks like this. It filtered for this Calanus genus, it added the sampling method column, and it only selected those 3 columns. All right, so just quick like plotting before I get into SDMs. So plotting in base R, there's this plot function, you specify X and Y for a simple plot. So we're just gonna plot some of these data. Not very many data points, kind of boring. But there are like better ways to plot, like within Tidyverse, for example, there's the ggplot2 package. And this, it just gives you a little bit more, it gives you like a little bit more flexibility with kind of like what you want in the plot and like aesthetics and that sort of thing. So we have our ggplot output. It looks like kind of similar to the base R, except you can like it's just a little bit easier to work with in my opinion. And this is an alternate notation of the same thing. But basically ggplot, you would start this by calling this ggplot function, and you would specify your data frame either here or in the genome. You also need this mapping equals aesthetic. And then this is where you would specify like your X variable, your Y variable, like your color or fill or anything like that. And then you would add on this geo point, which is telling ggplot to add points to the plot. Because otherwise this is just going to initialize like a blank plot. And this is going to add some error bars using geom error bar. This is more just to show that there are like a ton of geoms and just things that you can do with ggplot. Like there are a lot of resources online. If you want to do something specific, I'm sure there's someone who's done it before you. And this just improves plot aesthetics. All right, so finally moving on to species distribution modeling, hopefully that wasn't too boring. This is like what I do for my day job. Species distribution models are a really important tool in ecology for helping scientists kind of understand like the distribution of species when we might not have data that cover an entire area or time period, or if we want to like reconstruct past population levels, for example. And basically it just takes some sort of like species data, some sort of environmental data, and uses stats to compute an output, whether that be like abundance or probability of presence or something else. So I use this package, biomod2, and there are a lot of species distribution modeling packages out there. And I really, if you like want to get into this, I would encourage you to kind of like look around. But the reason I selected this one is because it's super easy to just like quickly get a bunch of different models running. So it's like pretty good for just comparing stuff. And there's also a super helpful vignette online on this like this biomod hub. So our first step, if we want to make a model, is to fetch our species data. And one way that we can do this is this open source data platform called Ocean Biodiversity Information System, or OBIS. We go to this website and kind of see what this looks like. So there's a lot of presence records, tons of data sets. So I wanted to look for a species like, I don't know, see what happens if I say turtle. Okay, I'm not very good at using OBIS online, but you can use it through this ROBIS package that we loaded earlier. And through this occurrence function, you can pull any species data that's available in OBIS. So for this example, I'm pulling right whale data because that's the species that I study, but you could like switch this species name out with like any marine species you can think of, and most likely there would be probably like at least one hit. And I'm using a start and end date just to kind of like put constraints on this. But we're gonna run this to load the right whale data. You can see it retrieved about 1,500 records. Sometimes if you like just don't have this start and end date, depending on the species, you can just get so many records that like your computer will kind of freeze. So next, we're just gonna plot our species data using ggplot. So I'm gonna load this world map from ggplot, which is just like a polygon that allows you to, like you can add it to the ggplot object. And then I'm just gonna run this. So this is plotting the right whale data with longitude, latitude, adding points, and adding the world map data, and then setting the limits on the map. Great. So here we have our map for our data. So we can see that it's mostly over in the US. We have this one random point over here in the Azores that we're going to ignore. And just for like ease of like just constraining the model again, I'm just going to select a bounding box that zooms in on the Gulf of Maine. And then I'm going to use all these dplyr functions to first like rename my columns so that they're a bit more intuitive. So I'm just replacing this decimal longitude and decimal latitude that came out of OBIS with just lon and lat. The year and month column in the OBIS data, actually. So this is like what comes out of OBIS. So it's this big object with 114 columns. You like basis of record, which is sort of like how the data were collected. Have like the taxa, date, geographic information. Yeah, just like all sorts of stuff. But it's like kind of a lot to handle if you don't like slim it down a little bit. But yeah, so our like the month and year from the right wheel data is a character, and so I need to cast those to numeric, which I'm doing here with dplyr mutate. And then I'm filtering just for the like the summer months because those are the environmental data that I have. And then I'm also filtering to this bounding box. And so when you're doing filters in dplyr, you can't, like, I think in Python you could do something where you had sort of like a greater than or equals like on either side of a variable, but R makes you like do each one individually, so you have to kind of add them up with this and, or if you wanted to do or, it would be that. And then we're selecting the relevant columns. And then we'll just plot again to see what our data are looking like now. And we can see that we've zoomed in on this smaller geographic region. So if we're going to produce a model, we need some sort of environmental predictors to figure out like what sort of relationships predict species occurrence, at least according to the model. And so I'm going to load some very basic environmental data. It is in a really unfortunate format where I like the extent and the names were not saved. And so there's definitely a better way to do this, which just shows that you can always learn stuff. But we're gonna load this like raster brick and okay. That does not exist. That's okay. All right. Fix this. So our environmental data does not exist where I had it, but this is okay, we can find it. So we're gonna have to create a directory for the environmental data. How you would do this is, or like a file path pointing to where the data are. So we're gonna create a file path. And then we can see, this is like the base directory right here where we are in this little files window. We go into, tutorials, like this is, I'm in Australia. Oops. This is where we were. So we're like quite a few levels in. And then our environmental data are all the way back here at the home level in the shared folder. Here. So we need to get to this file. And I think I'm gonna do like a very lazy thing and just select it, and I think it will work. Okay, so we're gonna create a file path. So this is gonna start with that for base directory, and then we're gonna share our we have this directory here, and now let's see if this exists. And it does, so that's good. So we can access our environmental data. And so we're just gonna change this following line here. to, it's gonna be a file path. There are many different ways to concatenate strings like this, but, and we're gonna have our environmental directory and our environmental data TIFF. Did that make sense to people just like looking the way that I explained that? And I just like having to point to a different directory. All right, so now we're gonna load this in. Hopefully it'll work this time. Okay, looks like it's working. All right, so we're gonna set the extent, which I pulled this from the original metadata. The variable names are depth, sea surface temperature, and chlorophyll. I have data from years 2005 through 2015. And we're gonna add the names. That's not going to work. Okay. Let's look at our environmental data. All right, so okay, for the rest of this, I'm gonna go through the HTML just because I do not have my data in the right place. And that is totally my bad, but we'll still figure this out. So this is what a rendered R markdown looks like. So this is all the stuff we went through. Yeah. Oh, yeah, so
Explain a raster brick, like.
yes, I totally can. So yeah, so environmental data can be like stored in a variety of ways. And this is arguably not the best way to bring it into R. But if you have gridded data, so like a .tiff or a like GRD or something, you're gonna need to load it with either like the raster package or something like Terra or stars, which are a little bit more advanced. And basically this will take in the gridded data and you'll have this object with like different spatial values and different data points at said locations. So I'm just gonna pull like one raster from this raster brick. So this brick is basically a data structure where you have multiple rasters together, which is why this is like a little bit more complicated than I wish it would have been. But this is just pulling out the first layer. And so you can see this is like class raster layer. The dimensions are this number of like rows, number of columns. This is like the geographic kind of, yeah, just like number of lat and long points. This is number of data points, and this is like the resolution of the grid, so it's like pretty fine scale. And then we have the extents. This is like the geographic mins and maxes, the projection system, also the source, and then this name, which is what is like tripping me up in this example here. But then if we were to plot this, yeah, so you see we have this like grid of, like it's along the US East Coast. And so it's a way of like storing gridded data. All right, so I'm just gonna go through the rest of this modeling example in this HTML. So this is like what a knitted R markdown looks like. So this is a really useful feature if like you were trying to, I don't know, like bring weekly updates to your advisor or something. Like I usually make an R markdown so they don't have to do the code themselves, but they can kind of click through and see what happened and stuff. So you can see these code chunks are rendering, and then we also have the output after each like operation where the output will be printed. So I'm just gonna skip through all this because we've already done this. Yeah, so getting to the species distribution modeling part. And as you can see, like this text rendered, so like if you guys like want to go back and like look at any of this, I've also put some links into like the different packages and different vignettes. So like if you wanted to go look at like the biomod documentation, it's all there. All right, so we've like fetched the species data, we've run this cell, we see our 1500 records. This is producing our plot here before we've zoomed in. And then we have this code chunk that was cropping the data and then plotting it again. All right, so here's the loading the environmental data component, which you can see that for this one, because the TIFF file was the correct shape, these names are assigned. And so we have like the first layer is bathymetry for the year of 2005. Then we have like SST, chlorophyll. This is like a climatological average for the summer months. And then it repeats for like 2006, 2007, et cetera. Obviously, like depth is the same across all the years, but it's just for consistency easier to have it like that. And then next, we're going to crop the environmental data to the same bounding box as we did our species data. And so we have this, this bounding box object, which just here. We look at our bounding box. It was this list of coordinates. And so if we wrap it in this extent function, which is from the package raster, it's going to turn it into a raster extent object. And then raster will basically just know how to crop this. Then I'm just going to plot the first layer to see that we have cropped it to these geographical bounds. And then just also plotting the SST and the chlorophyll. So it's always a good idea to plot your data to kind of see what they look like. In this case, this chlorophyll would make me think that maybe I should log my data when I put it into the model just because of how high the values are inshore. But for this example, I didn't do that. But it's always good to kind of look at your data, get an idea of what you're working with. So a super important thing to consider when you're building a species distribution model is what exactly you are modeling. So if you have species data, like what is it that you are trying to answer or like get out of this? So like, are you trying to model abundance? Do you want like a probability of presence? There are a bunch of different ways to go about this. But basically, because the OBIS data is of varying qualities, because it's from a lot of different data sources, it makes the most sense to model this as like a probability of presence framework, because we went into abundance, there would just be like all sorts of like uncertainty and stuff. And so we have presence data, which would allow us to do presence only modeling, but that is a little bit limiting in terms of what you're able to use. And so just for this example and being able to use like multiple algorithms and compare them, I'm using the biomod feature to create pseudo absences, which are basically like fake absence points that are sampled from the background environmental data. And so biomod knows to then go in to your environmental data layers and pick random points to use as absences. Like ideally we would have true absences, but this is again just for learning purposes. So here, we're going to add another column to the right well data, this presence absence. This is a binary indicator of whether or not a right well is present, or like whatever species you're using. So 1 indicates a presence, 0 would indicate an absence. And then we just need to format our training data. So when you're modeling, a lot of times you would set aside one portion of the data to train the model on, and then another portion for evaluation. And so here we're going to train on years 2005 to 2010.
I have a question.
Yeah.
So when it's absence, is it that you have been there and you have checked that it's not there, or it's just not being updated?
Yeah, so that's kind of a tough question. So ideally you would be sampling and you would know that the species is not there. And that would be sort of considered more of a true absence. But in this case, because I only have like the presence data from OBIS, I don't have true absences. And so I'm using this like pseudo absence generation approach. You could also like sample background points manually, or you could use like a presence only method like Accent.
And does always have something that they also report in some absences, or they just?
I don't think so. Yeah, I think it's just like occurrence data, because that would be really hard. Like it's kind of tough with absences because depending on the species and their behavior, like figuring out what that means is kind of tough. So like in the situation of like a marine species, like if you see them, it's probably because they're at the surface, and you don't see them, it's probably just because they're not at the surface. And so there's sort of like a a question of what exactly that means in different contexts, like different assumptions you have to consider. So we're gonna isolate that presence absence column
I have
for,
kind of a follow-up question.
yeah.
So would a pseudo absence just be like the lack of presence and you just didn't see?
Exactly.
Okay.
Yeah, and so like as you can probably guess, like that would have implications in like your final model evaluation and how you would use those results. Yeah, and so like I'm kind of brushing over a lot of this really quickly, but like if you're actually going about building an SDM, like there are a lot of questions you want to ask beforehand, and also probably just a lot of like model testing and selection that you would like go through. So Biomod expects like a specific format for its data. So it wants a column of training presence absence. And so we're just gonna filter for pre-2011 data. And then it also wants a separate column with the locations of that presence absence. So it's gonna be the lawn and lat. And then ideally we would also set aside data for evaluation. And so for this, I'm gonna use years 2011 through 2014. And Biomod has the ability to produce these pseudo absences for the training data, but I was not able to figure out if they could do it for the evaluation data. But what I've done is like a little bit of a workaround is just sample those background points myself from the environmental data. And so that's why we have this like background data frame. This is just taking that first like depth layer just to have the extent of the environmental data. And then it's turning it into a data frame with X and Y coordinates, renaming, sampling 30 random points, just because we don't have a lot of occurrences, we don't want too many absences. And it's giving them a PA value of 0 indicating an absence. And then this is randomly sampling before and after so that there's not like too. Let's ignore that. So then we're gonna like isolate our evaluation presence and absence columns. This is gonna be taking the right well data, binding the background data to it so that we have those absences in there with it. And then we're gonna do a deep flyer filter to select years greater than 2011 and less than 2015. So that's gonna get us into that 2011 through 2014 range. And we're gonna do the same for the coordinates. So we also need to subset our environmental data. There's definitely a better way to do this. But I've just used this like grep to grab like different layer names. So I just want like all layers pre-2011. And so this is just a little bit of a string select. And then I'm producing a climatological average of the contemporaneous variables, so the ones that change with time. So sea surface temperature, I'm averaging over all the years, same with chlorophyll, and then depth just stays the same because that doesn't change. And then we're gonna stack those together and add the names. Yeah, like rasters can be a little tricky to work with. And like I said, there are other packages that are a little bit further along, like Terra and Stars. And there's a lot of good documentation if you're curious about learning more. So this is just doing the same for the evaluation environmental data. Again, 2011 through 2014. And now finally, we're gonna format the data using a biomod function. So biomod's gonna take all those columns that you isolated, and behind the scenes, it will format that into a biomod data object that's like ready to put into the model. And so for this example, I'm gonna build a generalized linear model, a generalized additive model, and a random forest model. So these are 3 different algorithms that you can use. GLMs are probably the simplest, generalized additive models are a little bit more complex, and then random forests are a machine learning approach. And Biomod has like 10 or 11 different model options. And so you could do like any of these. And there's also for some of them like GAMs, there are options of calling different underlying packages. So like you could do a GAM in the GAM package or the MGCB package, for example. But this just shows like the flexibility of the package. So we're gonna format our data object. We have our response variable. We have explanatory variable, which is our environmental data. We have the coordinates here. These are the evaluation data. And then this PA.number.rep and the 2 below it, that's like the pseudo absence sampling approach. And so this is telling Biomod to sample pseudo absences 4 separate times. And so we're gonna get like 4 different models for each pseudo absence sampling, and so you can kind of look into the uncertainty a little bit. It's gonna sample 200 of them and use a random strategy. So you can see that our data are formatted, and it says here some data are located in the same raster cell, but Biomod has dealt with that because we have filtered raster as true. And we're just gonna look at the Biomod data object. So species name, right whale, 87 presences, 0 true absences, 795 undefined points. Just has some stats, evaluation data. And then these are the pseudo absence data sets down here. So there are gonna be 4 different ones. And like I said, like Biomod will create a model for each one so that you can look at like the range of uncertainty. And then we're gonna use this step to build our model. So we put the Biomod data object, which we just formatted, this modeling ID, which can be like whatever you want, the models that we want to run. This is gonna do a 5-fold cross validation. So you will get 5 different models for each algorithm, for each pseudo absence sampling. So again, like this allows you to compute uncertainty. This data split percentage is Biomod kind of splits like training and testing in addition to that evaluation data that we specified. This prevalence here is something you can play with. Like if you know that your presence points are more accurate or like more certain than your absence points, you can weight it more towards those presences. So currently this is just weighted in the middle, presences and absences weighted equally. But if I did like 0.7, then presences would be weighted more. And then valuation metrics, just like picked a few, because why not? We'll look at them later. Yeah, and then we're gonna run this. There's like not a lot of data in these models, so like some of them are just not gonna be happy because this is just not a very rigorous model. So we're just gonna pull out these evaluations here. So we have our model object, and it has a lot of features, one of which is these evaluations that we can pull from it. And you can see that there are a lot of models that were run. So we only had like our 3 formulas, but we just have a ton of models because there were all those cross-validation runs. And so for example, like we have run 1 of the GLM for the presence absence 1 approach. And then you have like all 3 evaluation metrics for that. So this is like a bit of a hassle to work with in my opinion. Yeah, just see how long this is. And so this is just a little hack to kind of get like what you would want out of that. And so I'm isolating the true skill statistic or TSS just because of the 3 evaluation metrics. This is the most rigorous. This is on a scale from -1 to 1, 0 indicating a random model would perform just as well as your model. And so if we plot this, we can see that all of our models are above 0. But these are still probably like not good models in any way. It just looks like they're fine. You can also pull out the variable contribution or importance. And we can plot that. And so for like chlorophyll, we can see that it's like pretty consistent that in the generalized additive model, it contributed about like 25% each time. But this also just shows like the range of model relationships that are determined in each run and why it's important to do a lot of cross validations. You can also do response curves, which will give you an idea of the ecological like plausibility of your model. So this will give you an idea of how your response variable, in this case, like probability of a presence of a whale, is responding to different values of your environmental covariates. And so for this one, this like does not seem very useful because this is just like not a good model. But if we go down here, these like look a little bit more interpretable. I'm not going to go a lot into this just because this is like a very simple example and therefore we're not really going to get useful results out of it, but there are a lot of resources online about like response curves and interpreting them. All right, so finally, we're going to project our models onto the final year, 2015, which we've held out. And so this is going to give us an opportunity to see how our model will perform on data that it has not seen yet. And so what we're going to do is pull out those environmental data for year 2015. And I'm going to make a projection for each modeling algorithm. And because we have so many different runs, I'm just gonna pull out the top 5 highest performing models based on TSS. So this is projecting the generalized linear model onto 2015 environmental covariates. The generalized additive models being projected, and same with the random forest. And so if we look at our projections, you're gonna get like 1 for each model run that you've projected. And this just kind of exemplifies how easy it is to just run a ton of different models across, like with a lot of cross validations, and then also be able to pretty quickly compare and visualize the results. And so here is the generalized linear model, and this scale over here, which I should have put units on, is from 0 to 1,000. Biomod saves the output on that scale because it is more computationally efficient. However, this actually represents a 0 to 1 probability of a presence of a whale. And so the way that you would interpret this is values that are higher, like this yellow would be like close to 100% probability of a presence of a whale in the summer. And then we can look at our generalized additive model, which we can see that this model is performing very differently from the linear model up here. And it's like just more smoothed across the board. And finally, we have down here our machine learning approach, which we can see is probably the most like ecologically sensical one. But again, like none of these models are at all good. But yeah, so depending on your problem, like you would want to consider like what algorithm makes the most sense. And then here's just a quick example of how you would like kind of access things in that projection object. So you can like pull out the projection itself, and then you would be able to like put it into, yeah, you just would have like some more plotting flexibility. So I've added like 2 data points from 2015, which again goes to show that this is not a good model. But yeah, so that's all I have for you. If anyone has any questions, I'm happy to answer them. Thank you.
So when you're like training your model and inputting your training data, so like for the environmental and occurrence points, what exactly is the model like getting from that? Like how is it training it? Is it like training it to show the most suitable areas for those occurrences?
Yeah, so with this like presence absence approach, what you're getting out of it is it's almost like a habitat suitability index is what you could describe it as. So it's telling you based on the model like determined relationships whether or not an area would be suitable for said species and like what the probability of that is. And so it takes in the environmental data in order to sort of find like correlations and relationships to the occurrence data. And then you're able to like project that across like the whole domain, like because we didn't have points for this whole domain, but the model has learned those relationships and is then guessing what it would think, like, or how high the probability is of an occurrence.
I have a follow-up for that too. So if you had like an entire data set for a species that's present globally, would you have to input that entire data set, or could you crop it to the area that you're trying to look at and then it can learn from that? Or like, would you have to use all of the occurrence points like ever for it to learn where it can be present?
Yeah, that's a great question. And that's an open question to an extent in modeling. So that's something called transferability, where if, for example, you were to crop that data set to a certain region, you could then project it onto a different spatial and temporal domain. But there are a lot of like implications of that, and sometimes it works, sometimes it doesn't, and you don't always know like if those relationships are static across the whole ecosystem. And so basically, if you were to have like a, crop it, create a smaller model, project it across the whole world, like you would just be assuming that the environmental relationships to the species are the same across that whole domain. And so that's something you also have to consider, is just like if that's a reasonable assumption, or if you think that it's like localized a little bit. And there are like a lot of really great papers like talking about the concept of transferability and like things you would have to think about. Thank you.
Question about time. Like maybe I missed like how is the time, like the seasons and all these kind of stuff.
Yeah,
Use that or?
yeah. So I did not really, just for the simplicity of this example. But so basically I used environmental data that was just averaged over the summer. And then I had like one, technically like one raster for each year, but then I also just averaged those. So I basically just projected onto a summer climatology. And you can get it down to the point where whatever resolution your data are, like however low you can go, like you could make like an hourly model if you wanted. Like there are ways to do it at like any time scale, depending on your data. But just for the simplicity of this, I just didn't want to like bring all that stuff in, and I already explained it quite confusing, I think, so sorry about that. Any other questions? Well, thank you.
Thank you.